Hello and welcome to part 3 of this introductory lecture on microbiology. If you've just stumbled upon this video, there are two previous parts to this. This video lecture, 3-part lecture video, is designed for an introductory biology class, high school biology class. Specifically, I'm making these videos for my 9th grade bio students. And so this is part 3 of a 3-part lecture on Intro to Micro. And we'll begin here with virus characteristics. We've looked at bacteria, some characteristics of them, how the body is able to adjust to bacteria, normal microbiota, how do bacteria make us sick, and now we're going to spend some time looking at viruses. Again, this is very introductory. There's so much more you could say about this, obviously, but this is just a survey of the major points that I like to cover with my bio students, just to give them an idea of microbiology and hopefully whet their appetites for it. So virus characteristics. Viruses contain DNA or RNA, but never both. And so that's what makes them what are called obligate intracellular parasites. So if you break that down, obligate means they must. Intracellular means within a cell. And they're parasites. They're parasitic entities. I don't want to call them an organism because technically I don't believe that they are, but they are parasitic, and the reason why is because of what this first bullet says. They have one nucleic acid or the other, but they never have both. And so in order for them to be able to make their proteins and carry out the process of transcription and translation, they have to get the other nucleic acid that they're missing. So they're going to have one or the other, but they never can have both. And in order for a cell to be a self-replicating cell, you have to have both. You've got to have DNA, RNA, and ribosomes, and viruses only possess one nucleic acid or the other. So they're relying on, or they're dependent upon, a host cell. And that's why they're called obligate. And intracellular means within the cell. So they have to get into a cell in order to get their required materials and chemical compounds. They have a protein coat around them. And that protein coat is what houses their nucleic acid. It's basically a shell around the nucleic acid. And this protein coat protects the nucleic acid, and it enables the virus then to, quote, survive or exist on inanimate surfaces for sometimes hours, sometimes days, and even in some cases weeks, depending on the virus itself. But that's unusual for it to be able to be viable for that long. Some viruses, and in fact many of them, have surface spikes, and these are projections that come off of the protein coat, which is called a capsid. These spikes come off the surface of the capsid, and they enable the virus, oftentimes when they are there, when the virus possesses a spike, to enter the host cell. So usually at the tips of these spikes there's a little bubble, so to speak, of enzymes. And so when they encounter a cell membrane, the spike opens up, releases the enzyme, and enables the virus to enter a cell. Or, going the other direction, the spike will also allow the virus to get out of a cell, which then enables them to go out and infect other cells as well. So these spikes are essential for the transmission of viruses from cell to cell. And they will also use the spikes for adherence, to stick to a cell membrane, and also to agglutinate cells or clump them up so that they can then enter neighboring cells very quickly and easily without being open to and being engulfed by macrophages or big white blood cells. Most viruses infect only specific types of cells in one host, and so whenever a virus leaves one particular type of cell, and I say the word leaves, I should say, or adapts and gains the ability to enter other types of cells, that becomes alarming. And this is where you get the CDC involved, or the WHO, or virologists and epidemiologists trying to figure out the transmission of this virus and what is the virus doing that's enabling it to now start infecting or entering a variety of different cells. Because the spikes that viruses have on their surface are very specific to host cell attachment sites. And so they kind of are like a hand and glove kind of fit, where the spike only can be picked up or attached to particular carbohydrates or proteins that are on the surfaces of host cells. Now, whenever they gain the ability to start entering other types of cells or even other types of or groups of organisms, that's when that makes them more virulent. The ability for them to spread increases rapidly as a result of that. And so whenever, for example, the flu is one that does that, and it can exist in birds, which is where the flu started, but it can also obviously exist in humans and also pigs as well, and other swine creatures like we think of domestic pigs as well. And so this gives the virus the ability to infect more hosts, which gives it the ability to spread more rapidly and quickly, which is what the virus is designed to do. I mean, that's its only goal: spread more quickly, get in as many hosts as possible. And so that's what we call the host range. How far can it spread out? What hosts can it affect or infect, and its ability to be able to spread. First isolated and studied way back in 1935. So this was, terms of years, the germ theory of disease was 1876, and so this was quite a few years after we realized, the germ theory of disease was, before that we were in like the dark ages of germs and bacteria and how they spread and how people get infected. We always thought it was just bad air we were breathing in. That's called the miasma theory, which is totally false and wrong, but it persisted for centuries. And then the germ theory of disease came along in the late 1800s. And so we're looking at 1935. This was just after the invention of the transmission electron microscope. And so they weren't able to, even though we had the microscope, it's still not powerful enough to be able to see viruses. So this is pretty early in terms of our understanding of viruses and how even infectious agents are spread in 1935. And it was in a viral disease called tobacco mosaic virus, which infects and kills tobacco plants. It causes their leaves to become all wrinkled up and mottled, and then they can't photosynthesize properly, and then the tobacco plant dies. So there's two types of therapy that I want to discuss here for a few minutes: phage therapy and oncolytic virotherapy. What are the benefits? So phage therapy, and I'm going to take these two separately, phage therapy versus antibiotics. So there is, as I mentioned in part 2, this antibiotic resistance problem among bacteria. How are we going to get around this? Because the bacteria developing resistance against our antibiotics way faster than we can develop antibiotics. It takes about close to 15 years for us to take an antibiotic from the mind of a scientist, so to speak, and then get it into the pharmacy. And that's way too long. Bacteria will adapt and they will mutate way faster than that. And so they will be able to devise ways to get around our antibiotics. And there needs to be a solution. Well, there is a very good solution. It's called phage therapy. And phage therapy is going on in the Republic of Georgia right now, not the state of Georgia, the Republic of Georgia, which used to be part of the Soviet Union, which the Soviets, or the Georgians, I guess I should say, they've been doing phage therapy since the early 1900s, very early, like 1905 early. And they continued phage therapy when the Western countries like the United States and Great Britain and so forth, they decided they were going to go with antibiotics instead. Antibiotics came on the scene in 1928 by the Scottish physician Alexander Fleming, and then it was mass-produced in World War II and given to soldiers of the United States and Great Britain, and it was insanely successful. Penicillin was like a godsend, and it eliminated and wiped out so many infections. Well, today, since World War II, we have been taking way too many antibiotics, and bacteria have adapted ways around our antibiotics and have been able to essentially not even, in many cases, be affected by them. And so we get more antibiotic-resistant mutants that are starting to come about. rather than the normal bacteria that were susceptible or able to be killed by our antibiotics. They are no longer able to be killed by them. And so we're getting to the point where it's going to be like a pre-antibiotic age, where you're going to get a bacterial infection, and then there's nothing. You won't have anything to take for it because the bacteria will have developed resistance against everything we have. Now, of course, that's not happening right now, but it is happening. We're in the process of this happening, and many scientists see this coming within the next 50 years, even, and in some cases, even less than 50 years. So a great alternative is phage therapy. It's been a very good therapy. It's been very effective. It's been tested for over a century now, and they're curing many diseases. They're clearing up many bacterial infections using phage therapy. What is a phage? I'll briefly explain what it is. It's a virus that infects only bacteria. So it's a very highly specific treatment. So let's say you get a particular type of staph infection. You can take a phage that has been developed to only attack and kill the staph bacteria. Antibiotics are non-selective. They kill anything. If it's a bacterial cell, the antibiotic gets rid of it, good, bad, or ugly. But with phage therapy, you've got a weapon that hunts down and destroys a very specific target. And so the good bacteria survive. So you don't lose all of the beneficial bacteria that are part of our normal microbiota. It's only targeted to get rid of that pathogenic bacterium. And then once all those bacteria, the pathogenic ones, are gone, the phages are then, they're done. They don't replicate anymore. And so their numbers just dwindle down to nothing. Within the body, and they can't infect our cells, so they're no harm to us. Phages can only infect prokaryotic cells. Phages cannot infect eukaryotic cells. They only infect bacteria, and that's it. So there's no danger to us. At first, they were given by injections, and many with phage therapy. That's the way it still is today, but they're starting to develop pills now so that you don't have to have a needle. And with blood-borne diseases, there's no risk of spreading infection of these diseases now. You can take a pill. The phages get released into your bloodstream, and they basically search and destroy that particular pathogenic or disease-causing bacterium. And it's a great alternative. The United States was also doing it along with the Soviets in the early 1900s. We left phage therapy with antibiotics, and we instead put all of our money in research and development into antibiotics and not phage therapy. The Soviets continued with phage therapy, and they have not left it, and they've continued to develop it and research it, and they're doing very well with it. If you want to look it up, Phage Therapy Center. It's in the Republic of Georgia. You can Google that and find out more information about phage therapy. Great alternative to antibiotics. Oncolytic virotherapy. What that is, and my bio students should know this, onco means cancer. Lytic means to burst or to split the cell. So this is a type of therapy that bursts or splits apart cancer cells using viruses. And so it's kind of along the same lines as phage therapy. Now, what's our current mode of treatment for cancer? It's chemo and radiation. Now, what makes a person deathly ill in many cases? Chemo. So let's get rid of chemo, a type of treatment and modality that we've been using to treat cancer for almost a century or a century by now. Let's get rid of that and move into a new age of cancer treatment, which we should. I mean, we've been doing chemotherapy for a century, and at first that was it. It was wonderful. It was a great way to treat cancer. Chemotherapy was kind of, I don't want to say stumbled upon, but it was almost accidental in finding out how these chemotherapeutic drugs and chemicals actually can kill cancer cells. And it worked really well. But the side effects, chemotherapy, like antibiotics, is non-selective. So in the case of cancer, and you take chemotherapy, these drugs are designed to kill any rapidly producing cell. So that's why your hair falls out when you're undergoing chemotherapy. That's why you have nausea and vomiting and skin lesions, because all of these rapidly producing cells are being slammed by the chemotherapeutic drug. And so people get violently ill in some cases, deathly ill. They are so nauseous and they're vomiting and they dread the next chemo treatment. And if you've ever had chemo treatments, I feel badly for you, and some of you maybe have experienced some of these effects. They're awful. Just I don't know firsthand, but I have seen. I've had relations. My family relations have had chemo treatments, and it's a rough thing to see somebody have to go through all of that. So, what's an alternative? This oncolytic virotherapy, where the viruses specifically target and destroy cancer cells. Now, it's not that easy. I'm generalizing very much, but that's the basis of how this works. And virotherapy also trains or teaches the immune system to recognize these transformed or cancerous cells that are within the body. So you get this kind of double benefit where the viruses are killing the cancer cells, but at the same time, it's also teaching the immune system to find these transformed and cancerous cells and kill them. So a great alternative to chemotherapy. Phage therapy, great alternative to antibiotics. Oncolytic virotherapy, great alternative to chemotherapy. This is where the Western countries, the United States, needs to be putting its research and money and development into these two therapies and starting to phase out those old, outdated, dangerous therapies that we're using now. I mean, with antibiotics, we're creating a global crisis of antibiotic-resistant bacteria. So anyway, there you go, some education for you. Look it up if you want to learn more. There's plenty. Are viruses alive? So I kind of touched upon this earlier, that outside of a host cell, they're inert. They can do nothing. And so that we classify them as non-living. They have few or no enzymes of their own, so they need their host cell enzymes in order to begin producing more of themselves. So it's kind of like they're a virus producing factory, but there's no electricity to the factory. They can't produce anything because they're lacking the electricity to turn on all the machines. So they need the enzymes from their host cells. They need the electricity. They need that voltage to go into them to enable them to start mass producing more viruses. So when they do reproduce and they make more of themselves, they cause infection just like any other living thing. So I guess you could say, in that way, they're kind of the same as bacteria or algae or fungal spores that cause disease or something like that. So they're like other pathogens. You could probably classify them as a very complex mixture of chemicals or a very simple living organism. I tend to lean more towards a mixture of chemicals because, as I said when we started, they have DNA or RNA. They don't have both, and they can't carry out protein production. And so what good is it as an organism? It's just sitting there. So I kind of lean more towards a complex mixture of chemicals. So some anatomy of the virus. Virion is a complete, fully developed infectious viral particle. It's got everything ready, and it's ready to infect. It has a protective protein coat called a capsid, which will enable it to enter cells and to clump up with other viruses and so forth, and to clump cells together too. So this capsid surrounds the nucleic acid, whether it's DNA or RNA, one or the other, never both. Some viruses have an envelope that surrounds them. It's a lipid and protein and carbohydrate conglomeration. It's a complex that covers the capsid. And part of this envelope many times contains the host cell membrane, which enables it to just slide right into a cell. It kind of tricks the cell into bringing it in through endocytosis, and then the virus can get into the nucleus and start making more of themselves. So the envelope is not, it doesn't surround all viruses, but a lot of them it does. And so that makes transmission and the ability of the virus to get into the cell much easier for the virus. So COVID has an envelope around it, and that envelope then allows it to gain access into our respiratory cells quite easily, and makes its way into there. However, if a virus does have an envelope, it makes it very easy to I don't want to say kill it, to inactivate it, because viruses that do have envelopes, they have to have the envelope. If they don't have it, they're done. They can't do anything at all. And the envelope is made of mainly lipids, and lipids can be broken down easily by alcohol. And so that's why it's so important when they're saying, when you're trying to prevent the transmission of COVID, for example, wash your hands. Well, soaps are good at breaking down the lipid components of the envelope. Alcohol works great as well. It can break down the lipids too. So in one sense, This makes COVID weak, but COVID it spreads incredibly rapidly. It's highly contagious. And so it's getting out there and transmitting itself very quickly and rapidly, even though it does have an envelope around it. But this gives it access. So it's kind of like it's got pluses and pros and cons to it, pluses and minuses. Yeah, it has an envelope, which makes it susceptible and pretty easy to neutralize, but at the same time, the envelope enables it to easily enter into cells quickly, rapidly. And a spike is a carb-protein complex, and we use these spikes to identify the viruses. So, for example, we classify the flu virus like H1N1, H1N2, H2N1, and so on. And so you have all these different naming designations for the flu viruses. And the way we name the flu viruses and classify them is because of their spikes. They have H spikes and they have N spikes on their surface. And then virologists have just called it H1, H2, H3, and so on. And they've done N1, N2, N3, and so on. And so they have a number of H spikes, different H spikes, and they have a number of different N spikes as well. So just looking at viral anatomy, some of them are just a helical virus, like the tobacco mosaic virus on the left. It's just a cylinder with RNA inside of it. Some of them are called, the next one over, that would be a polyhedral one, which has many sides to it. And within that then would be the nucleic acids, an adenovirus, as you can see there, and you can see some of the spikes sticking off of its surface. You have other ones like the example of the flu. It's got a membranous envelope around it, so an enveloped virus as well, with the spikes sticking off of its surface. And then finally on the right you have what are called complex viruses, because they have a complex shape to them. They got a bunch of different stuff going on, and a bacteriophage is a classic example of a complex virus. So they come in three, usually you see three, in this case you see four different types of viruses in terms of their shape. And that's kind of a general shape classification to them. Relative sizes, I just put this slide on here to show you they're small. So you've got a human red blood cell there in the corner. It looks like it's a giant sun or something. Compared to the viruses that are next to them, viruses are incredibly small, and they need to be in order to gain access into cells and to gain access easily. You can see even next to the E. coli bacterial cell, they're small even compared to that as well. So viruses are tiny, very small entities. So how do they reinfect? So you get the flu, for example, once. Well, you can't get it again. And the reason why is because of the mutations that occur to what are called their antigens, and it's called antigenic shift or antigenic drift. Shift and drift. Genetic shift, genetic drift, you'll hear that as well. In this particular case, we should refer to it as antigenic shift because it's the antigens, or in this case of the flu, the spikes. That's what is what's differentiating one flu virus from another. So these spikes, these antigens, can mutate easily and rapidly compared to other viruses. So you may have produced antibodies to one influenza virus, but the virus mutates pretty rapidly and easily, and so that can infect you again. So your immune system recognizes that particular spike. The immune system is very specific in terms of our secondary and our specific immunity is what I'm trying to say. Whenever we encounter a particular virus, we produce very particular antibodies against that virus, against the antigens that are on the virus. But then if those antigens change on the virus, then we have to go through a whole new round. It's like it's a brand new infection. It's still a flu virus, but the surface has changed. So as far as our immune system is concerned, yeah, it's the flu, but our immune system is like, whoa, this is a brand new antigen. And so it has to produce a whole new set of antibodies, and that takes time to do that. It takes like 2 weeks, sometimes even more in some cases, to develop the antibodies necessary to tag all these viruses for destruction. Antibodies don't kill. The antibodies will tag antigens for destruction by some other immune system cell. And there's a whole variety of those which we won't get into. So you get it once, you may get it again. Does the flu vaccine stop it? And why do we have to get the flu vaccine year after year? Well, you don't have to, I guess I should say. But why is the flu vaccine offered year after year? Like, for example, you had, I hope, you had your measles, mumps, and rubella vaccination when you were a child. You don't have to get it again. We're done with that. We get our rounds of vaccinations of it when we're babies and young children, and then we're done. There are no boosters given. You don't have to get a new round of them. They don't have to keep, meaning pharmaceutical companies don't have to keep making new measles and mumps and rubella vaccinations. Why? Well, those ones don't easily mutate like the flu does. So they have to offer, or they don't have to, again, the pharmaceutical companies offer new sets of vaccinations every year for what they believe is going to be the new or the predominant flu virus that's going to be circulating among people that particular winter. Now, it's not a guarantee. They offer the flu vaccinations beginning somewhere, around when a little before flu season starts, around October. You can start getting your flu shot if you so desire. But that flu shot is not a guaranteed vaccination against the predominant flu virus that is going to be circulating among people. So it's not a guaranteed vaccination. Now they have a lot of mathematical models and there's equations that they go through to make this prediction, and the reliability is pretty good. I mean, we can trust math. Math is great. But we can't know for sure because influenza can change and mutate so easily. So speaking of vaccinations, what is it? So a vaccination or a vaccine is a prepared biological agent of some kind, and it's usually a weakened organism, like a weakened virus, or it's bits and pieces of a bacterial cell, like when you get your whooping cough or pertussis vaccination. That vaccine is bits and fragments of the pertussis bacterium, and then it's put into an injection form. And then you get that injected into your arm, say, for example. And then what those little components do, the weakened viruses or the bacterial parts or the proteins from a bacterium or some inactivated toxin from a bacterium, what they do is they stimulate an immune response, and then you develop antibodies against those things that have been introduced into your body, but they're in a weakened form, and so you're not going to get sick from it. At least you shouldn't, and in many cases you don't. They're very successful in manufacturing vaccines that do not cause the disease. Then you develop antibodies against it, and then when you encounter that particular pathogen later in life, you can rapidly produce antibodies against it later, and your immune system takes care of it and wipes it out. So there's 3 good videos here. The virus video, that one is, the first one is by NPR, and it shows kind of simply through computer graphics how viruses spread. History of vaccination is about smallpox, the first vaccine that was ever successfully administered in 1796. 1796, Edward Jenner, he was way ahead of his time. And it gives a little story on that. That's a TED Ed video, which I love. And the best TED Ed video is pandemics. So go to TED Ed and type in pandemic. We're in the middle of a pandemic right now, so I recommend you check it out. It's a great video. It doesn't include anything about COVID because the video was put out before COVID, but it does mention SARS, the first round, SARS-CoV-1, which was in 2003. We're dealing with SARS-CoV-2 right now. So basically it's kind of the same virus, but it's different. It's mutated. So anyway, those are good videos. I love the pandemics ones. That's a great video on TED Ed. So how do they reproduce? Well, there's 2 main ways, lytic cycle and lysogenic. Lytic cycle, the virus replicates, fills up the cell, boom goes the cell, and it bursts. And the death of the host cell, and the viruses get released, yeehaw, and they can go out and infect other cells. Lysogenic cycle, the viral DNA incorporates itself into the host cell DNA. So it doesn't kill the host cell, but it's making more of its instructions. Every time the host cell divides, the viral DNA divides with it. And then what happens is that the cells will leave the lysogenic cycle, enter the lytic cycle, and start reproducing and rapidly making more of those viruses, and then the cells burst, release the viruses, and go infect other cells. So it's not just one or the other. Sometimes it can begin with lysogenic and go into the lytic cycle. and then leave the cell and then go and infect other cells. So lytic viruses leave the cells. Lysogenic, the viruses do not leave the cell. The instructions for making the viruses keep getting replicated over and over and over again, and then they can go into the lytic, and then it's mass infection. So this is kind of what it looks like. It looks kind of complicated. You start in the left side with the red number 1, and you can see how that cycle, the red cycle, just goes around, and then the cell bursts, and the viruses get released. Or, if you go to the number 1, you can go down into the picture in the middle, and then head down to the lower right, where you have the DNA incorporating itself into the host cell DNA, and then the host cell just keeps reproducing and making more and more and more of the viral DNA. And then it can, like it says there in the blue number 5, it can then begin or initiate a lytic cycle. And then you can, it's like a figure 8, and you can see how then it can leave the cell and then go and infect other cells. So lytic and lysogenic cycles, 2 ways that viruses will reproduce in cells, bacterial cells or eukaryotic cells as well. Viruses and cancer. So I want to talk about this because anything that viruses alter nucleic acids within the cell, they mess with the nucleus. And so anything that alters cellular DNA has the potential to make normal cells cancerous. So those would be oncogenes, and anything that begins with onco means or refers to cancer. Several types of cancers are known to be caused by viruses. About 10% of all cancers are, the causative agent is a virus. That's as far as we know. It could be more. There's still more research and understanding we have to do with this. We're not sure about all of these and the connections between the viruses and the cancer. And in some people, it causes cancer, and in somebody else, it doesn't at all. So there's a lot of genetic factors, individual genetic factors that are involved in viruses causing cancer in people. But we do know of some that are causing cancer in many people. This was the relationship between viruses and cancer was first demonstrated in 1908, a while ago, over 100 years ago, with chicken leukemia. So they were doing research with chickens. There was a lot of research on chickens and poultry that was going on in the early 1900s, and we learned a lot from them in terms of immunity and the immune system. This was one here with chicken leukemia, viruses and cancer, and immunology. It all kind of goes together because the immune system, of course, is involved in taking care of cancerous cells. So there was a lot of research going on there at the turn of the century. Oncoviruses are viruses that can transform normal cells and make them cancerous. Oncoviruses and transform cells or cancerous cells, their rate of cell division, they hit the pedal to the metal and they just divide without restraint. Loss of contact inhibition. Normally, under normal circumstances, cells will run into each other and touch, and when they do, they will stop dividing. With transformed cells, they don't care about that. There's no personal space. They just keep dividing and piling up. And the normal cell cycle has been disrupted as well. So the virus causes the cells to make more of themselves because that's what the virus wants. It wants more cells. It wants, specifically, more nuclei so they can get more DNA. And so sometimes viruses cause cells to become multinucleated, when under normal circumstances they only have one nucleus. They want the nucleic acids, and so they will cause this to happen. And then the oncogenic viruses, their genetic material gets integrated into the host cell DNA, and so then that leads to more of them being produced later on because the instructions for making them gets incorporated into the host cell DNA. So every time the host cell divides, so does the instructions to make more viruses. There are, in terms of infections, and maybe this would be more of like a clinical aspect or speaking in terms of healthcare, there are 2 types of viral infections, latent and persistent. So latent, and that's what the word means, it's kind of like it's latent, it's hiding, it's not showing itself. So the virus remains an asymptomatic host cell for long periods. So asymptomatic, you have symptomatic, so you're showing symptoms. Put an A in front of it. No symptoms. So the host cell is infected. It's got the virus in it, but it's showing no symptoms of that. So cold sores caused by herpes virus, shingles caused by varicella or chickenpox virus. Oncogenic viruses do the same thing as well. There's no sign of cancer, but then suddenly it appears because that's how they work. They incorporate their DNA into cells. Cancer doesn't show up, and then once it starts turning on the machinery for making more of the viruses, then the cell begins to transform and become cancerous. Many remain latent in nervous tissue, like the case with shingles. So if you've had chickenpox, you have the pox viruses in nervous tissue in your spinal cord. And then later in life, shingles can appear. It's the exact same virus that gave you chickenpox. But they're not. It doesn't come on as strong because you have partial immunity against chickenpox because you had chickenpox. But the shingles appears in a limited local area of the body, and shingles is very painful. I had it just a few months ago, and a couple of months actually, and it hurts. So my chickenpox was reactivated, and it exhibited itself as shingles. So for I don't know, over 40 years, it was hanging out in my spinal cord, and then it became activated. And why did it? It's hard to say. Am I immunosuppressed? I don't think so. So there must have been some other factor, maybe my age, I don't know. But anyway, it came back again. And so that's a latent viral infection. A persistent is one that it persists, it stays, it progresses over a long period of time, generally fatal, and years later that disease will appear. And the disease is often caused by another virus. So there's a connection here with measles and what's called fatal encephalitis. So that's swelling of the brain, and it's uncontrollable. And about 1% of measles cases ends up with fatal encephalitis. HBV and liver cancer. So hepatitis B virus, the hepatitis, refers to the liver, hepatitis B virus, and liver cancer appearing decades later. Human papillomavirus, which there's like 200 of them, and they cause warts. Well, there's a few strains of the papillomavirus that can cause cervical cancer, and it's persistent. It progresses over long periods of time. So it's not bad, but it will gradually and slowly get worse and worse. What about plants? How do they get infected? They do. But they're generally protected by the cell wall. So viruses typically cannot enter plants because the cell wall is so thick and strong. It's made of cellulose, and it's a very stable protective structure around the cell. So really the only way they get in is through wounds, like the break of a branch or a stem, or insects that are sucking the plant's sap. So the insects are infected with the virus. They then transmit the virus into the sap of the plant, and then it just circulates through it. It's very similar to like malaria, where a mosquito bites you, transmits the malarial parasites into your bloodstream, into your sap, basically. So the insects will inject the viruses into the sap of the plant. It circulates through the plant, and it can cause disease in the plant. Viroids are infectious pieces of RNA. So notice the name oid, it ends in oid. And so they're like viruses, but they're not viruses. Potato spindle tuber disease is the number one viroid plant disease. These viroids are responsible for millions of dollars in crop damage every year. And what it does, you can see the leaves there of tobacco mosaic. I'm sorry, that's tobacco mosaic virus causing that mottled appearance to the leaves, which then interferes with photosynthesis, making them incapable of producing their carbohydrates, and then they'll die off slowly. And the last topic that I want to talk about are what are called prions. And prions are infectious proteins. So they're not even close to being living. They have no nucleic acids at all, not even close to being a living thing at all. First explained in 1982, and we first saw this in sheep. And what happens is these prions are proteins that infect normal cellular proteins, and they cause the normal cellular proteins to become an infectious protein. They bring them to the dark side, so to speak. And so what happens with prions is they slowly but steadily convert these normal cellular proteins that would be on, say, the cell membrane. And these proteins are used for transporting substances into and out of a cell, or moving neurotransmitters, or ion channels, or something like that, charged particles in and out of a cell. These proteins then become transformed. Their structure changes because they have been infected by these proteins that are called prions. It's very strange, and there's a lot to learn about prions. It's unusual. Like, how do they do this? How do they cause a normal protein to transform? Many questions about prions that are unanswered right now. But one by one, these normal proteins will change. And as a result, you lose the function of the protein, and that's going to lead to cellular death. So they're inherited, they're transmitted by ingestion, transplant, surgical instruments, and they can be spontaneous. And what does that mean, spontaneous? Well, they appear for no explicable reason. There they are. And so we don't know where they came from. Can we treat prion diseases? We can. It's kind of, it's not very successful, but we can treat them with enzymes or NaOH, which is sodium hydroxide, a very concentrated base, in an autoclave. They're resistant to biocides, they're extremely stable, they're resistant to denaturation or the changing in their shape, so they stay in that infectious protein configuration and they remain that way. Many are believed to be inherited, and there are 5 human prion diseases. That's the case with them. They are inherited and passed on from one generation to the next, and they are fatal. There is no cure for a prion disease. If you are diagnosed with a prion disease, it's a death sentence, and unfortunately people that are afflicted with them will not survive. But fortunately, there aren't many people that get infected with prion diseases. But there's one called Kuru, which was a result of cannibalism, eating the brains of deceased relatives. So the relative died, and the tribes people would ritualistically eat the brains of their deceased relatives, I guess in order to get their wisdom, I don't know. But then they ended up getting the prion disease, and so there's one, fatal familial insomnia is an inherited one. Creutzfeldt-Jakob disease is another one. So there are the ones that get passed on. What we deal with around here and where we live is chronic wasting disease among the deer population. So that's a disease that has been spreading among the deer population across the United States. It's within the elk, mule deer, and whitetail deer, at least. So that's a prion disease that's spreading among that population. And the letters that we use, the PRPC, that's the normal cellular prion protein. Those are the ones that we have that are normal and doing their job. And then you have the PRPSC, which is the scrapie protein. And scrapie was the disease, the prion disease that was discovered in sheep in the early 1980s. These prions accumulate in brain cells forming plaques, and the plaques are these just dense areas that you see, like, for example, in Alzheimer's too. So we think there might be a connection between prions and Alzheimer's. We're not sure, and the plaques that are associated with them. The only way you can know is by doing what's called a post-mortem. So you do an autopsy and you look at the brain tissue under the microscope, and you can see what it looks like under the microscope, and it gives this spongy appearance, and so they sometimes we call it spongiform, where it looks like a spongy brain. Those white or lightish colored circles and so forth are not supposed to be there. That's not usual for brain tissue. So these are characteristic of prion-affected brains. And then here's on the left a section of a normal brain, and you can see the section of an affected brain by a prion disease called Creutzfeldt-Jakob disease. It just destroys brain tissue. And you can see how much of the brain tissue has been destroyed by this infectious disease. So the cells just start to die off. They can't do their job because the proteins change, and then they're not receiving nutrients or they're not transporting things properly. They can't carry out their normal function, and then they just start dying off, and you get these, in a microscopic view, you see the small holes, but macroscopically, you can see these giant holes in the brain tissue. Video on the left, that just is a good video that explains prions, and then the video Fatal Familial Insomnia gives it's a story about a family that this disease runs in, and it's kind of a personal story about this family. So 2 good videos to watch related to prions. So that ends our discussion. That's the end of part 3 of this 3-part lecture on microbiology and introduction, and hopefully you've learned something valuable and it's given you a better understanding of microbiology.